Test, test. All right. Cool. You'll be doing all of this when you present, so be ready. How many of you know how to record lecture? You haven't done that before? Never? Not conference presentation? Really? Wow. You should start doing so. You're doing masters or PhD, so be ready for the market. Yeah, this is a very important skill. You could be very good in science, could be very good in doing research, but there are something else you should do. So for example, if you want to be a professor in university, normally there's 40% of load of teaching, there's 40% of load for research, and there is 20% for services. So if you fully focus on research, you'll miss some stuff on the other side. And then your evaluation will be bad. You may not make it for the tenure track, right? So be careful about that. Try to enrich yourself with the skills aside from research. Research is important, how many papers you publish, whether they are good venues and so on, right? It's very important. But also you need the teaching skills. Unless you are going for research positions, we still have in academia like assistant professor or research assistant professor, they call them. That is 100% of research load, okay? But you could also do it in industry, where you go with R&D or research groups. Most of the companies, they have research groups. So you could do research, but still part of research, going to conferences to present. So you can imagine the loop. You will need the presentation skills anyway. Even if you build a family, you raise a kid, right? And you want to teach them something. You don't understand your language. So you have to really be good in presentation to convince or convey those messages, right? So in one way or another, you still need those presentation skills. And part of it is to record your talk, because you will grow up your audience rather than just focusing on, we have 20, 40 students in the courses, and then you could target larger groups, right? All right. So last time we covered about 3 topics related to pre-alignment filtering. Those are very important filters. And in the assignment number 4, you're going to implement one of them, okay? So I will give you the freedom to choose one of them, then present it in your own, and figure out how much optimization you have done to the sequence alignment or the dynamic programming because of the use of pre-alignment filtering. This time we're going to cover what we call advanced topic, which is a full read mapper published this year called Genome-on-Diet. But this is a quick reminder for assignment number 3. So be careful. Last time we released it, and you have, I think, about 5 days, okay? The deadline is really sharp, so pay attention to that. We are not going to extend it. Right after it, we're going to release assignment number 4, but I give you a bit of relax. If you don't want to submit it on April 7th, you have up to April 14th to submit 3 and 4 together, okay? But the earlier you do it, the better for you, because after that, we're going to fully focus on the review paper, okay? So don't delay it, otherwise you'll be in trouble. All right, hopefully by now, everyone agrees that we really need genome sequencing in order to read the genomic data. So until now, we cannot read the full genome. What we have to do to send the genomic sample to one of those genome sequencing machines in order to give us the data, right? And the throughput of those machines really vary. So we could have, this is PacBio, you know, the fridge size, you still remember the PacBio? This is producing reads of length normally from 20K all the way to 30K, okay? As we call it, PacBio. We have 2 types of reads over here. We have the short. Hopefully you still remember all of those. And we have the long, right? The lung, we have two types. We have CLR, and we have circular consensus. Okay. Or this is the old name. Can replace the circular consensus with the hi-fi, which is high fidelity read. Okay. We didn't cover this one because it's a bit old. This is without circular consensus, so you just read the read once, you sequence it once. But this is circular consensus. You go at least 30 times over the same genomic sequence to improve, keep improving. So every time you go over the circle, you read chunk, you read another chunk, you read another chunk, you keep reading, and you pick all of those pieces together and build consensus, which is majority voting, for example, or alignment. And this is short, similar to Illumina. This is very recent technology coming from PacBio. And we have very different machines for each of those sequencing technologies. Okay. And in this machine, we have the ONT, Oxford Nanopore, right? And here we have Illumina. This is one of the highest throughput NovaSeq devices. This is the highest throughput you could get in Illumina. So you can see really the throughput is very different, and the sequencing time to get the sample. So this is the throughput you would get, and this is the sequencing time. So apparently, this sequencing machine, what gives you is the raw genomic data, is the FASTQ file, basically. And you still need the computer to do some processing, filtering, and pre-processing as well for all of this data. However, those are specialized machines. They are just built for the things that they are supposed to do, which is just sequencing, okay? But those are general purpose, nothing customized for sequencing or genomic data, just general purpose computer. That's why normally we have those as very fast. You can see the throughput much higher than what we have in processing power in typical day general purpose machine or computer. That's why those are slow. So when we have a closer look at the compute infrastructure we normally have, which is the SSD, the basic storage, right? Where we store the FASTQ coming from the sequencing machine. And then we have the main memory, then we have the processors, right? Whenever we want to process something at the CPU, we cannot jump directly to the input data or the IO. We cannot have something coming from the IO directly. We have to push it to the SSD all the way to the main memory, and then to another level of memories, we call them cache. So we have different level of caches from L3, L2, L1, and then we push it to the CPU. So the more we move toward the CPU, the less the memory we'll have. So normally here, about 1 terabyte of data, right? Here, the DRAM normally 32 gig or so, right? Cannot go to terabyte, right? Unless it's server class. So from 1 terabyte to 32 gig, the cache is around kilo or megabytes, okay? Very small, very little. However, the more we move toward the CPU, the faster the memory is. That's why it's very expensive to have large capacity. So the DRAM here is much faster than what we have here, but less storage capacity, okay? Likewise with the caches, since it's very close to the CPU, we have to accommodate some data in order to have the different power moving the data and the computation power to cope with each other, okay? Normally, between those components, different components, we want to communicate data from here to here to here. We have some communication links, which are normally wires, right? And those have sometimes limitation in terms of the bandwidth, and as well as the power, the energy. So it's very expensive to move data from here to there to be processed at the CPU. And this is what we call a data movement. So moving the data from one component to another to another. So data movement is a major bottleneck in modern computer architecture. And if you're interested in this topic, I would recommend you to go over this paper, which was trying to analyze the Google Chrome workload. So you have many tabs in the Google browser, right? And you try to figure out what kind of effect it has when open a tab, close a tab, or opening many tabs, right? So try to compress those tabs all together. And this is already applied in today Google Chrome. This is very old paper, but we start seeing it only recently. Google already applies such technique. Once you are absent from one of the tabs, right, for a very long time, it will be offloaded, right? And once you visit again, it will be loaded again, okay? So it will clear the main memory utilization for that tab if you are no longer reusing it for a certain time. And once you visit it again, it will start loading it again to the main memory, start processing it. This way, they save a lot of computation power as well as the storage in the DRAM. So over 60% of total system energy is spent on data movement. You can imagine about 30% or 40% just spent on the CPU to do the processing, and the rest is just moving the data. There is no computation at all. Just moving the data from the storage, DRAM, caches, CPU. Just try to wait for the data to be coming all the way through very long journey from the SSD or the main storage all the way to the CPU, and then we process it. So that is giving us kind of motivation by reducing the amount of data without losing accuracy, without losing the result that we are looking for. We could improve the overall computation or overall energy, the overall benefits in terms of memory and so on. So the problem is currently with the data. And in genomic, the data is already huge. So a single genomic file, FASTQ file, coming to be processed, at least is 15 gig. That is at least. So it could go over 10 terabyte. For example, this is real data that we could get from ONT devices, which is the raw signal. So this should be converted into FASTQ, right? And before converting it, the raw signal data is already huge. This is has two formats. First one called FAST5. The other one is SLOW5. Okay. So these size, these kind of data files already normal. This is the norm for genomic data. Okay. So now this amount of data need to be moved to SSD, assuming the SSD can fit, or those files can fit in the SSD, and then moving such a huge data, waiting for the data to be processed, and then come back, store the data, and move on. That's already a lot of resources. And what we have in this paper, a very recent paper published 2 months ago in Nature Communications, called Tamming Large-Scale Genomic Analyses via Sparsified Genomics. And the key idea here is that we normally have the genomic data, whether it's coming from sequencing machine or cloud storage, such as SRA or GenBank or ENA. Do you still remember those two names? The SRA and ENA? Hopefully you use them for the assignments. Do you remember what they are? Any thought? Excellent. So this is the SRA, which is all the kind of FASTQ file you're interested in. If they are publicly available, you mostly find them on the SRA. Okay. So the SRA is a huge database that accommodates all kind of publicly available FASTQ. So any research center or hospital can upload those files. Okay. They have some dictates on those files, type of data and so on, but you still can submit for free. The ENA version it's very similar to the SRA. However, this is American and this is UK. This is Britain, okay? Or European, all right? So this is hosted in NIH. Hopefully, it's still there, okay? And this is hosted somewhere else. Most of the data you will find in both of them, okay? So this is out of connection. Normally it's happened that you cannot connect, you cannot download data. You can visit the exact same accession number or the ID in the other database. But there are still some data unique to this. There are still some data unique to that, okay? It depends on who uploaded. the data is mine. If I upload it to that database, done. Then the others don't have it. Okay, but sometimes they share the access between them. Anyway, so assuming you are getting the data, regardless how you get the data, you could also simulate it as well, generate it yourself, store it somewhere. But then in traditional method, you would move it to the SSD, right? And then build what we call an index, right? So that's the first step we learn in this course, which is indexing. You would like to index. Why we index the data? Why we index the data? Think about you have a book, right? You have a book and want to read. Do you read it all? You're interested in a certain topic. What you would do, you go to the preface or the index of the book, right? Go to the topics, try to pick the topic of interest, try to figure out what is the page number that the topic starts with, and then go to only that chapter or section and start reading it, right? This is why we have indexing. Indexing is a very fast way to retrieve or visit any portion of the file without loading the entire file into the memory, right? So we build an index, which is a way that tells us if you are interested in this portion, start from here. If you're interested in this portion, start from there, okay? And so on. But does not tell you what is inside that portion, okay? Done, just index portion of this huge data, just small portion snapped from here and there, right? And load them in the index data structure. So we build the index and store those indexes inside the SSD. Once we need them, we move them to the main memory, all the way to the CPU, and we start processing them. However, you will see that in practice in the assignments, as we move forward with assignment number 3, you already have the index built from assignment number 1, right? And now you want to move it to assignment number 3, so you already load it, you already store it somewhere, but you still need the FASTA file, you still need the FASTQ file. Even if you build the index, you still need those 2 files, plus the index itself, right? Which is the hash table. Why you still need the FASTA file, although the hash table is built on top of the FASTA file? So this represents that file, right? That is one. The second thing, once you move to sequence alignment, what do you need? What do you need to run sequence alignment? You need a pair of 2 sequences, right? So you need a pair of 2 sequences. This does not give you the subsequence of the FASTA file. Although you detect subsequences or pieces of the genomic sequence, or those seeds normally, right? We call them seeds, are already matching. And we know that this subregion should match. But we don't have the entire subregion. We have seeds in that subregion. This is what the hash table is giving us, right? Just words or those subsequences coming from the entire segment of the reference. What we do, once we detect what we call a mapping location, and this term, I already defined it in assignment number 3, so be careful to that, okay? Once you detect the mapping location based on the matching seeds coming from the hash table, what you would do, go to the FASTA file at that mapping location and extract a subsequence of length equal to the read itself. This is how you get the pair of genomic sequences. Once you get the pair, you just do sequence alignment. As simple as that. So at the end, you still need all these files in order to do read mapping, right? And that's the traditional way of doing it. Now, what this work tried to propose is that don't build the index. Don't build the index, okay? Get the FASTA file, right? Try to sparsify it. Sparsify it. What does mean sparsifying? Sparsifying is that make some choice, not random choice, okay? Systematic approach, systematic choice of choosing what data is important to you. So think about You have some data, right? You make some choices, not random, okay? You said, I want every other k-mer to be considered. Every other 10 characters, okay? 10 mer. So you choose the first mer, you ignore the second. This is the FASTA file, the genomic sequence itself, okay? Ignore one, you consider one, ignore one, consider one. Okay? Someone else says, oh no, I want to consider every other two, okay? So the choice is up to you. You choose whatever you want to ignore portion of the file, okay? And that's what we call sparsifying. So instead of having dense data, right, you just make it sparse. By having a sparse data, you convert this picture into that, right? We have now less data to be processed. So think about this. How much you save by ignoring every other line? Half. So now if this is 10 terabytes of data, becomes 5 terabytes. That's a huge saving. Think about how much we process out of that file, the indexing, okay? Now over there, you could continue whatever you are doing, okay? You could continue processing this sparsified data without losing accuracy. You're not losing data. You're not losing data, okay? Let's see it in practice, but hopefully you capture the idea behind this word. Now, we've already been visiting this figure many times, right? And this is what we call a brute force. A brute force meaning you try every single possible ways, okay? Assume you have, for example, 10 ways to solve a problem. You are trying all of them. Hopefully, you will come over the best solution over some time, okay? That's a brute force. You are trying every single solution. And a brute force for read mapping is basically to revisit or visit every single location in the genome. Hopefully, you will capture the region that is similar to the read. But we already said that this is very expensive. What we've been doing is that indexing, as we discussed, capture some seeds, store them, build the hash table, move on. There's no Genome-on-Diet. There is no sparsifying over here, okay? That's traditional method of doing it. And we already see some different methods. Those are state of the art. However, the index size could be huge. We are using a genome, a human genome of size 3.2 billion character, 3.2 billion character. However, index is much larger than what we have, at least 2x, right? We have here sometimes 3, 4, maybe more, 5x of this size, right? Here we have about less than that, maybe 1.5 or so, but the indexing time is huge compared to this, for example. This is 7 gig, which is 2x over this, and the indexing time is much less than what we have over there, okay? Now, once we are doing profiling to see the motivation, why we should worry about indexing, right? We try to profile different applications, okay? We got one of them is read mapping. One of them is read mapping. How much indexing contributing to the total execution time? How much indexing, once you index the file, how much it contributes to the total execution time? We find that it's about 10% to 27%. So 1/4 of the execution time dedicated only to index the genomic data, okay? Of course, if you do it once, store the hash table somewhere, done. Then it won't contribute significantly to the execution time. However, it still contributes because you are going to read the hash table sometime and do queries to the hash table. But still, it won't be worse than that number if you do the query only. But if you build the hash table, it's going to take that much of the total execution time. So if you do something over the indexing and you try to improve it, you're going to improve at least 1/4 of the execution time, okay? When we check other application, for example, metagenomic taxonomic profiling, which is an application that tries to figure out what kind of microbiome do you have in a sample. Okay, you have a sample, you collect it from environment, and you try to figure out, do we have viruses? Do we have bacteria in it, right? And that's kind of a very important application for indexing, because you have a huge amount of data. You want to compare every single read in the FASTQ file to a huge database of microbiomes. And we already discussed about this. If you still remember, we said a very challenging puzzle. You remember the figure where we have the puzzle, right? The pieces. And we couldn't know what reference genome, what reference picture we should compare with, right? So we use several pictures. And then we try to compare those the puzzle pieces to all of those pictures, because we don't know what we have in hand. Is this FASTQ coming from COVID? Is it coming from E. coli? Is it coming from other microbiome? We don't know, right? So we just compare to everything in a database. And when we check those kind of application, we see the indexing contributing to 97% of the execution time. This is really tremendous. So if you do any contribution in indexing over there, you're going to slash the execution time, okay? Now, the Genome-on-Diet goal is to reduce the execution time and memory storage footprints of genomic analyses via sparsified genomics. So what we would do, we want to look, instead of having the entire reference genome, we replace it by a small portion of the genome. And instead of looking for the entire read, we are going to consider only small portion of the read. Hopefully, by using those much smaller data, we could figure out the right location where we could do the comparison. And this is, by the way, very similar. If I give you a sentence, a very long sentence or a paragraph, right? And I ask you to find it in a book or a research paper or even one-page PDF, what you would do? If I ask you to find a complete sentence in a PDF, not a word, a complete sentence, what you would do? Control F, right? You activate the find window, and then you start typing the first character, the second, the third, the fourth, right? You're not providing the complete sentence. You start providing portion of the sentence. And hopefully, you will find it right away, okay? Now, let's see a real example. This is what I always like about the algorithm. And hopefully, you are considering that in your presentation. Whenever you're trying to present an algorithm, please focus on how it works, okay? Don't show the algorithm. Don't show the pseudocode. Don't do that, okay? Show me how it works. Just provide an example. Give dummy data or toy example and show me with some animation, with some cartoon, and so on, okay? Let's see it in action. Assuming you have a reference subsequence, this is a small portion of the entire genome. Assume you have a read sequence, okay? That is an exact copy of that. There's no difference, okay? Exact match, all right? I decided to consider every other character. Every other character. So keep one, ignore one. Keep one, ignore one, right? And I have those copies as a result. If I try to find this one using this query, will I be able to find it? They are still exactly the same since I applied the exact same pattern over those two. So I keep one, ignore one, keep one, ignore one, right? So that's still exact match. Okay, that's nice. And this greatly, as you said, reduces the execution time, storage memory footprint by 2, because I slash the size by half, right? So replace 2 by 4, by 6, by 8, by 7, but by how many numbers you try to ignore, right? Which is your pattern. But there is a problem. What is it? Now, going back to our example, we want to keep the exact same reference, the exact same read, They are exact copy. I'm not going to do any edit operation, no edit, okay? However, what I'm going to do, okay, ignore one character. This is not edit operation. What is it? Do you remember the sequencing machine? It's not always going to give you a read at certain location. It just randomly reads your genome, randomly. We never have any guarantees where the read would start. The reference subsequence could come at location 10, at location 5, right? But the read could come at any location. And that is randomly sampled or subsampled from the reference genome. So now assume the read is not starting from the first character, it just starts from the second character, right? Just hiding this character. And apply the exact same pattern. So you consider one, you ignore one, consider one, ignore one, consider one, ignore one. You do the exact same thing over here. Now, can you compare the green with the red? No single match. Nothing. Nothing. No single match. Even the k-mers. Look at all the characters. Even if you extract the k-mer from here, no single seed match. You can imagine that. They are exactly the same thing. We just ignore one character. So this is not an easy task to sparsify the genomic data. You are going to have sometimes nonsense data. There is no match at all. And then you flag this as does not exist, but it already exists. So what is the workaround? How you could solve this problem? So I had those ideas for some time. So we started developing this algorithm around 2022, 2021. And it's giving me a hard time to come up with a solution to this. We try to have an easy workaround. However, you might have more dedicated algorithm to solve it. Let's see the workaround we have. We said before doing anything, we are going to consider a k-mer out of this data, okay, which is specified version, assuming that we are going to consider the first character, ignore the second. First character, ignore one, okay? Just keep one, ignore one, keep one, ignore one. And that is the first few characters that we would get, okay? But we also consider the other case, which is ignore the first, consider the second. Ignore, consider, ignore, consider. Over here, we consider, then we ignore, okay? Over here, we complement what we have over there. Why we do that? Why we do that? Because we are not sure whether the read that's coming from the sequencing machine would always start from the first location or the second, right? So we are considering all cases, assuming that, what if we start from the first character? Okay, we have it covered. What if the sequencing machine starts from the second? We have it covered. But wait a minute. What if the read starts from the third location? What if it starts from the fourth location, fifth, sixth? This is a repeating pattern, right? A repeating pattern. If the read starts from the first, it's already covered. If the read starts from the third, it's already covered. If the read starts from the every other character, we have it covered. We have it also in the complementary locations. So if it's covered in the second, it's already there, the fourth, the sixth, and so on, right? So it doesn't matter if it starts, as long as we have a regular pattern, which is 1, 0, 1, 0, 1, 0, 1, 0, where a 1 means consider that character, a 0, ignore that character. Then everything is fine, right? All right, so we still didn't decide which one to pick. We have two choices. Either we go with this sparsifying pattern, or we go with that. Which one to pick? We check both of them if they exist in the reference genome. Apparently, does this exist in the specified version of the reference genome? Yes, we have a hit which appearing in the very first few characters, right? This is a hit, right? However, if you check this, we don't have it anywhere. Not even at the end or in the middle. We don't have it at all, right? So apparently, this one is leading to a correct match because it exists in the specified version, and then we move ahead with the entire complete version of that one. Remember, what we do here, we just check small portion of the data. We don't need even to specify the entire read. We just specify small portion, and then we try to compare and see if it exists or not. Now, you could have some outliers or some challenging situation, which is in real life, normally we don't have such nice example. What if both of them exist? What will happen? We continue moving with the next k-mers. So we extract the next, this is 1, 2, 3, 4, 5, 6. We extract the next 6 characters, right? The next 6, the next 6, until we make a decision which one to pick, okay? And normally, after a few of them, by practice, after 5 k-mers, you could make a final decision, okay? This is in practice. You could try it yourself. You will see after 5 k-mers, the versions will start being totally different. This version over that version start being really different, okay? So after capturing 5 k-mers, you could make that decision which specifying pattern you are going to choose. All right, so after we make that decision that we are going with this pattern, then we got this version, and then they are matching again, everything is nice. Now what we gain after we do that, as we discuss, we slash half of the size of the genomic data. How we do it in practice, how we do the full read mapping. What we discuss currently just indexing, right? We didn't discuss read mapping. We just discuss how we compare genomic sequences. But what if we have very large FASTA file and very large FASTQ file? So normally we solve that issue using 5 steps. Compressed indexing, pattern alignment, compressed seeding, location voting, and then sequence alignment. So how is this similar to what you have in the assignments? Compressed indexing, you already have it in assignment number 1. Number 1, you were just reading the FASTA and FASTQ file, right? But it's part of the solution, part of the first step. But the actual thing that is similar is assignment number 2, right? In assignment number 2, you were asked to index the FASTA file. Remember, we don't index the FASTQ file. We index only the FASTA file, but we do seeding to the FASTQ file. And there's a difference between indexing and seeding. Please remember those terms. However, what is new here is the compressed thing, okay? In your assignment, you're not doing compression. You're just doing indexing, right? You don't have this step. It's not part of the assignment. It's not part of traditional read mapping. What you have after indexing is directly seeding. And after seeding, you are doing the location voting, which is assignment number 3. And I give already a large portion of the credit to your innovation to build something new for finding the mapping location, okay? Hopefully, I will find some creativity in the answers. Try to come up with your own method on how you detect what kind of subsequence from the FASTA file that I should compare the read with, okay? Because you need for sequence alignment to have a pair of sequences. One is the read already given, but what is not giving the other partner, okay? Which is the subsequence coming from the FASTA file. How we detect that, how we know we should have that subsequence over other subsequences, okay? And this is what I'm looking for to see in your answers. Sequence alignment is already common, okay? You already have it in assignment number 3. All right, now let's see how we will perform a read mapping using sparsifying approach. First step, compressed indexing. What we have here, a genome sequence. It could be 3.2 billion character as in a human genome. It could be virus, could be anything. That's a FASTA file. We have here the characters, and those are the index of, or the indices of those characters. Okay. Remember, in computer science, 0 is a thing. Okay. In computer science, 0 is a thing. So we should, in principle, start from 0, not from 1. Now our pattern, hopefully you don't need this. Let's erase it. The pattern, it's up to you. Okay. For me, I want to use it as 1, 0. And this is the minimum repeating pattern. Okay. So it's up to you to choose it. If I choose 1, 0, what does it mean? If the length is 10 characters, it means my pattern, remember, this is the minimal repeated pattern. Means this is the minimum, cannot go less than that. However, if the size of the genome is 10, then I'm going to repeat it 5 times. 1, 2, 3, 4, 5, right? That's 10 characters. So if I ask you what is the minimum repeating pattern of this sequence, 1, 0, not 1. What is the minimum repeating pattern? If you choose 1 and you repeat it 10 times, it will be 1, 1, 1, 1, 1, 1, 1, 1, right? This is not that. The minimum repeating pattern is the pattern that is minimum, cannot be less than that. And if I repeat it, it will be the same thing. So 1, 0, 1, 0, 1, 0, 1, 0. Now, if I ask you for that, 1, 0, what is the minimum repeating pattern of that? 1, 0, 0. Excellent. 1, 0, 0, right? That's the minimum repeating pattern. It doesn't matter if the genome is 100 long character, just repeat it. Okay, 1, 0, 1, 0, 1, 0. We got this. Now, if it is not really fixed or a multiple of those size, for example, if we have 9 character, just ignore the last one. If the genome is 9 character instead of 10, just ignore the last one. Then you have 9 characters over there. Hopefully, this is clear. Okay. Now we apply that pattern to the genome, and what we got is this version. Okay, this copy, we got it from the genome itself after applying the pattern. So whenever we have 1, we consider that character. Whenever we have 0, we ignore that character. And then we have A, C, T, G, C, T, A instead of that. Now, just continue doing whatever you were doing in assignment number 2, which is indexing. Just choose the method you like, extract k-mers out of that sparsified version or compressed version, and then do whatever you want with all of those. Do you remember how many methods we studied for indexing? Can you name 1, 2, 3? What is the one you use in the assignments? Minimizers. Okay. Oh, okay. All right. I like that. Minimizers of minimizers. Okay. What else? What did you use? Okay. What did you use? Sync merge. Okay. Excellent. All right. Yeah. So you can see that we have different flavors. Whatever you like, you could apply, right? So everything else is really normal. Whatever you were using for indexing, you could apply over here, right? And just store it in a hash table along with the start location. Remember, always indexing involves start location. All right, now step 2. This index, you can save it somewhere. You can, whatever you do with it. Now, the next step is to process the FASTQ file, right? So we got the first read. Remember, reads are coming from sequencing machine. Sequencing machine has no guarantees on where we could extract or sample the reference genome. We could sample it at any location, randomly, really randomly. If you have exact replication, the exact same genomic sample, okay? You make it a copy, you have 2 samples now, You hand it to the exact same machine, you sequence both of them, you will get different FASTQ file. Okay? It could be the exact same content, like the read are the same, but could be shuffled, right? One after another, one before another, and so on. So what we would do, since the pattern is regular and the minimum repeating pattern is 2 characters, then we cannot have more than 2 choices. If the minimal repeating pattern, 2 characters, we cannot have other than 2 choices, right? Which is either you have it as 1 0 1 0 1 0 or the complement 0 1 0 0 1 0 1, right? So now we will consider the first choice. Just apply 1 0 1 0 1 0. This is what you got. You build the k-mers. You will see over here that we have certain number of k-mers, but we could have 4, right? As a previous example. Why? Because this k-mer still is not spanning the entire thing. So we could have 3rd and 4th. Now we consider the other complement version, which is shifted by 1 step, which is 0 1 0 1 0 1 0 1. We got another copy, and then we extract the k-mers. As I said, the number of k-mers we extract from here is gradually increasing. We don't have to extract all of them at once. We extract a group of them, and we use them to make a decision. If we could make a strong decision saying, no, I'm pretty sure that this is not the right pattern, that's the right one, then done. You don't need to continue. And this makes it really fast, because once we try both of them, that is a duplication of effort, right? Which might spend more time rather than saving time. But if doing that minimal job over here, and by extracting only this number of k-mers, right, you really don't need to apply the entire pattern to the entire sequence. You could just apply the first 5 characters or so, extract the k-mers, done. Now, what we do next? We already considered the 2 choices. There is no other choice. You could try yourself if you don't believe it. If you want to read starting at this location, we still have that cover and that cover in here. Okay? So regardless of the location, it's already covered. However, we might have a worst case when the pattern is not regular, okay, or it's not 2 characters. Think about in here, the minimal repeating pattern is 3 characters. So how many times we need to consider those choices? 3 times. Either I can consider the other color and the blue. Is this the first time to use blue? So we don't need this guy. We could have it 1 0 0 1 and just repeating itself, right? Or could have 1 0 0 1 0 0 1 0 0, right? So if the minimum repeating pattern is 3 characters, then we could have 3 choices. Either the pattern will start from the first character, or the second, right, as in here, or the 3rd character. Everything else is repeating itself. Won't matter. If you think it starts at 5th character, you will find it in one of these options. And the problem is, again, is not with the pattern itself. The problem is with the sequencing machine. I'm not sure at which location it will start reading. That's why I'm considering these 3 options, right? Okay. Now, hopefully, we understand how we do that. Now, what we do with the k-mers, we check if they exist in the index, okay? So those do not exist in the previous slide. You can see if any of the k-mers exist here or not, okay? We don't have them. Those exist. Then what does that mean? It means the use of that pattern increasing the chances of having those as exact match. So 2 is greater than 0, then we are going with the shift amount of 1. If we are using 3 options, then we have a shift amount of 0, here 1, here 2, right? So the shift amount could be any of those 3 choices. Now, the 3rd step, once we make the decision of what amount of shift we could use, we go back to the read itself, the exact same read that we were using here, okay? We just have it, another copy, and now we apply the correct shift amount. So we decide with which one of those 3 we should go. We choose one of them only, and then we apply to the entire thing, and we extract the k-mers. Okay? I should have add 2 more k-mers over here, because now we have to, not it's up to us, we have to extract all k-mers, okay? So that we can use them for the indexing or querying the index. Now everything else is exactly the same as what you are doing now in assignment number 3, which is here I choose location voting, okay, which is something similar to the hint I provide in the assignment. So you have a hint, right, saying that it's up to how many seed matches you would get accumulated in one region. If they are up to or above a threshold, it means this region is good in the FASTA file. And then I have to pick that region and start comparing with it using dynamic programming. Okay? So now this is the read. This is the read sequence, and this is the reference genome, right? If I see a lot of seed matches scattered around certain region in the reference genome, this is chromosome 1, this is chromosome 2, then I would consider a threshold. Are those seed matches above a threshold? They're a number in quantity, okay? Is it above a threshold? Yes. If so, okay, this is potentially a good mapping location. Then I'm going to grab this region, the subsequence, and start comparing it with the read sequence, build whatever you like, pre-alignment filtering or sequence alignment and so on. There's another region, another candidate where the seed matches, their number is really exceeding a threshold, then I would consider that subsequence as well, and that subsequence and so on. We have different category of those mapping location. We have primary, secondary, and supplementary. We don't have to go over those. I'm not asking you in the assignment to find them, right? You are good if you have at least one mapping location, okay? But what I ask you in the assignment is to find all those mapping locations. You don't really need to classify them into primary, secondary, and supplementary, okay? As long as you could figure out what is the right subsequence in the FASTA file that share large number of seed matches, and by large is above a threshold, okay? As long as you could find all of those, then you are good to go. The final step is just to apply any of those algorithms that is able, where's the blank? That is able to find 3 things. Do you remember what are the 3 things that we need from any sequence alignment? We need really 3 important things from any edit distance algorithm or sequence aligners and so on. Remember, our ultimate goal is to find the genetic variation that causes a disease, right? So finding them first, we want to find how many? How many variation or edits, right? And then the next, what are they? Okay? What are those edits? The third thing, what are they? Okay? And by they is edits, right? What do you mean over here? We have 4 choices, right? Or 3, because the 4th one is not considered an edit. You can consider it edit if you like, which is we have substitution, right? We have deletion, we have insertion, and we have a match. So where are they? At which location they are happening? What type we have? And how many of them? Do we have 5, 6, 7, right? To answer all those questions, we really need sequence aligner. In the paper, we choose one important implementation of sequence aligner, which was very recent at that time, and it's the fastest. So we choose that one to be used in our method. So in summary, this is exactly the steps that you need to perform. First, you choose a pattern, you apply it to the reference genome, you get a compressed version of it, and then extract the seeds normally, store it somewhere. And then the second step, you would have the pattern, try to make whatever possible choices. So here we have a shifted version of that pattern, and then we try to apply it to the read sequence, get the compressed version, try to guess which one of those possible options are best. Here we have a lot of occurrences. Then we said, okay, probably this is the good one. Now the time to apply whatever we decide over here in here. Remember, here we extract few seeds, but here we extract now all of them. And then we use them to query the index. And then we try to make a location that is best suited for the sequence alignment. Here we use location voting, means whatever most of those votes, each seed match considered as a vote. The higher the votes on a region, the better that subregion for us. Then we extract that subregion at location 15 because start from there. We pick next few characters that's equivalent to the length of the read, and then we try to do the comparison. We got out of it a SAM file or a BAM file. Okay. Now, hopefully this is clear. There were some other contribution as part of this work, but those are optimization. Okay. For example, you build SIMD implementation, make it much faster. You implement it in C, okay, not Python. If you go with Python, you cannot compete with high performance tools. Okay, so you have to go with C. The lower, the better. And moving from C, you could also leverage the CPU itself. What kind of capabilities you have in the CPU? If you have SIMD instruction, then it would provide much higher performance. Now it's a bit challenging because Apple is moving to its own processors. 2-3 years ago, Apple was using Intel processor, right? So you still can apply the exact same instruction, which is SIMD, which is relevant to the processor. If you change the processor, those instructions won't be available. These days, we have the M4, right? M4 processors coming from Apple itself. And then we don't have those instructions, but we have alternatives. We have alternatives to those which can act as SIMD instruction, but it's different way of using them. So you have to learn how to use those instructions for that processor, and then you could apply SIMD instructions. There are many other optimization. You could go over it in the paper, which is not relevant for us for now. But there is one important point which I get when I present this work. Okay, when I go to conferences and present this work, normally I get this as a question. What is the difference between Genome-on-Diet? And by the way, you can figure out why we call it Genome-on-Diet, right? Because we are trying to lose weight. We are trying to lose more character, to not consider them, right? And then consider only small portion of it. What is the difference between Genome-on-Diet and spaced seeding? Spaced seeding. What is spaced seeding? Let's see. So in spaced seeding, which is things that you are doing, there is no difference. Just extract all k-mers. But then we choose a pattern, okay? Whatever you want, choose it. You could choose the exact same pattern you use for Genome-on-Diet. And that pattern you apply to the seeds themselves. You don't apply it to the reference genome. You apply this pattern to the extracted seeds. So you make the extracted seeds smaller. For example, this is 1 triple zeros, 101001. This is not regular, okay? This pattern is not regular. And then if you want to have it minimal repeated pattern, and you want to decide which one to use, how many times you need to apply the algorithm? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So you have to consider 9 times of shifts. What if it started this location, this location, this location, this location? So 9 times you need to apply that. But still, this is not expensive as we discussed, because we are going to extract only a few number of seeds every time we apply the pattern. Now let's see, when I apply this pattern to the first seed, so 1, we consider the first character, then we ignore 3 characters, we consider next character, ignore the A over here, consider the second A, and then ignore 2 characters, consider the last C. And this is how we got all those seeds, okay? But in Genome-on-Diet, Now I want to apply the exact same pattern we choose over there. We just repeat it over the entire genomic sequence. This is exactly the same. So I apply it 3 times. I got the specified version of the genome, and now we extract the seeds. Here I'm using a minimizer, same way as I'm using here. Or sorry, not minimizer, even just all overlapping k-mers. There is no minimizer here, okay? But the type of seeds that we got out of the overlapping k-mers, can't compare them. They are totally different. First, from the same region, from the very same region, very same number of characters over here, here we extract only 3 seeds. The exact same region. I'm not ignoring any characters. Exact same number of characters. I'm extracting only 3 seeds using Genome-on-Diet. But in space seeding, I'm extracting how many? 6 of those seeds. And you can see some benefits and some downsides, okay? First of all, those seeds are very nice because they allow for some variations, okay? Now, if I want to match a read with a genome, and there are some variation between the seeds, I still can match them, okay? Because I have gaps. And that's why we call it space, because I have some spaces. And within that space, so if I have here T in the reference genome, here as a T, I still can match that one with the one with the T in the reference genome, because those 3 characters are ignored. So I don't care what they are. They could be anything, right? So in this way, I'm increasing the number of matching seeds, right? So that's way we use it normally. If we couldn't find a lot of matches, we could allow for gaps, and then we could allow for more matches to happen. But here also we could do that. Here we are ignoring some characters, and whatever mismatch is happening over here, I can still capture them, okay? But here, again, we have less number of seeds. That means less execution time, because I'm going to query only 3 times instead of querying 6 times the index, right? And then the location voting would be faster because I have smaller number of k-mers to be checked. And that's going to reduce peak memory, storage, and so on. So the index of this would be half the size of the index of that. So it's not only about the number of k-mers, but also when I store 100 instead of 200 k-mers, or 1,000 instead of 2,000, that's going to reduce the index size. Then it's much smaller. The last point is very important. The last point is very important. What is it? All seeds have the same pattern. All seeds, we use the exact same pattern applied to all of them. Okay, all of them, even this 1, triple zeros, 1, 0, 1, 2 zeros, 1, right? But in here, can you figure out what pattern we use for the second? For the first one is clear, which is the exact same pattern over here. But the second one is triple zeros, 1, 0, 1, 2 zeros, 1, 1. Okay, 1, 1, not single one. The third one even is much different. 1, double zeros, double ones, 3 zeros, 1. So every time we generate a seed, apparently we'll have a totally different pattern if the pattern itself is not regular. We're not repeating the minimum. Okay, so for example here, 1 triple 0, 1 0 1 0 0 1. That looks not regular, right? So to have it clear, I prepared this example for you. So now, in this case, it's easy. So you can apply 1 0 1 0. Okay, if this is the pattern that we choose. And then if you check the first seed that you could extract, it will have this pattern. If you check the second seed, it will have the complementary pattern. Or it can be the exact same pattern we use here, but shifted. So you can ignore the 0 here and have it at the end. Doesn't matter. So both of them you can consider as having the same pattern, or each one has different one. Okay. Now, if we use a different pattern, for example, 1 0 1. So that is the minimal repeating pattern, 1 0 1. Now the first one will have 1 0 1 1 0 1, but the second one 0 1 1 1 0 1 1. Okay. Now we have an extra one over there. Now the larger the pattern, the minimal repeating pattern, the different, or start seeing new pattern applied to the seeds. For example, 101, 00, 11. Over here, it's totally different than what we have here. 00, 11, 01, 001. Now, why is this important? Why I should care? What if I have different pattern or the same pattern applied to those seeds? Why I should care? Any thought? Any idea? No? Okay, we said in the previous example first, for example, we have A, C, 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 T, A, C, 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 okay? Now, I want to extract the first seed, which is A. Don't care, don't care, don't care. Then we have 1, 0, don't care, 1. And then don't care, and 1. Okay, this is the first seed, right? Now we said we could match, this is a read, and this is a ref, A, C, T, C, T, A, C, C, C. So in principle, I just apply this variation. Let's have it in red. Okay, I just did single variation, and this is like in practice. Normally, we don't expect the read to match exactly with the reference because there are variation between me and someone else. And then if I apply the same pattern, I want to use A 3 times T exactly as above, right? And then 2, and then C. So does those match? Yeah, both of them match each other, right? So perfectly fine. Then space seed helped me to find them. Now, what if this, or let's keep it. What if this and this are different? Now, this will be different, right? And this is don't care anyway, right? It doesn't matter. This is don't care, but this is not don't care. This is C. Now, when we match this, do they match? No. So now space seed fail, right? Because the number or the location of don't care position is fixed. So if the matches appear to happen at some other location that they don't care, okay, or that is not don't care, then we won't get seed matches. But now the chances here much higher to have a match between any of those seeds because we don't consider a fixed location of don't care. Okay, they are not fixed. So every time here we have totally different pattern. That's increasing the chances of having more matches between the seeds. Okay, so even if those variation are hard to predict, like could happen anywhere, we could still capture them, increasing the number of seed matches. And remember, the sequence alignment, the dynamic programming, even if you use the most sophisticated one, the best algorithm out there, if you miss the seed matches, you're not going to do anything. Done. You lost that location. Then you are not going to cover or discover the variation that caused the disease because you just missed it. Okay? So it's all about seed matches. The more seed matches you have, the better the chance that you are going to discover that location in the reference genome, and then the better you are going to do with the sequence alignment. Okay. So we have some experimental results. We try 3 different applications: read mapping, containment search, metagenomic profiling, and we got some results. You can see the numbers, you can read them in the paper. However, you can see the benefits, it really varies. Here we have single order of magnitude or less. Here we have 2 orders of magnitude. Here we have 2 orders of magnitude. And the reason, if you remember in the motivation, when we profile, the contribution of indexing to the total execution time was different from one application to another. And read mapping was barely, at most, 27%, right? But in metagenomic profiling, we have it about 97%. Okay, so it depends how frequent, how often, how important we are using indexing. If it's really important, major portion of the execution time, then the benefits that we would get much higher because we are reducing the index size, and anything rely on the indexing, it's going to benefit directly from such kind of algorithms. We also try different window size for the minimizer. The k-mer here is fixed, the k size, but we try different window size all the way from 5, 7, 9, 11, 13, 15, 17, 19, and we can see some points where is the best, and sometimes is not the best. The red point here, what does it mean? This is the default configuration of Minimap 2. Minimap 2, we already emphasized it too many in this course, which is currently the state-of-the-art tool for read mapping. So if we want to use read mapper, or for example, if you are curious, you want to compare the performance of your read mapper after, for example, assignment number 3, you're already having a read mapper result, right? So if you want to compare your result with existing read mapper, you would go with Minimap 2, try to install it on your machine, run it on the same FASTQ and FASTA file, and then compare the results. This tool will give you the mapping location, will give you the cigar string, will give you the distance, and so on. Okay, so you could use that. And the default, if you try it, this is the performance that you would get. This is the peak memory. We have the execution time. I don't know if it's considered. Yeah, I forgot to include it since we already have the execution time here. But we have in the paper, you can see this is G. So we have A, B, C, and then D, E, F, and then all of those. So you can check in the paper and see. But the point here is that the default of Minimap 2 is not always the best. The default is not always the best. You can see even the peak memory is different with using different window size. Again, the window size is relevant to the minimizer algorithm. If you use something else, then you don't expect to compare with it. But if you are using minimizer, you can compare with it. But you can still have it as a ground truth. Okay? You can still have the SAM file coming from this tool as a ground truth for you to find the best location of every single read coming from the FASTQ file. You can see here Genome-on-Diet compared to Minimap 2 in terms of number of variation, how many variation you can detect. Okay? You can see sometimes we perform better, sometimes no, we miss something, and so on. The source code, the paper is available in the archives, so you can access it freely. You can get the source code from GitHub and play with it. This is the paper, published 2025. That was our advanced topic for today. Do you have questions? All right, so what we cover so far in this course, we cover a lot of topics about indexing, seeding, and then pre-alignment filtering and sequence alignment. So we cover all aspects of read mapping, right? So we've seen this advanced topic relate to the full read mapping. We already invite you for another advanced topic in our department, right? For advanced LLM for genomics. And moving forward, we have some time to have some advanced topic. So either I'll send you email that we will have at this particular day another advanced topic, or I might suggest some videos that you could watch, okay? But until then, that would be hopefully the last lecture for this month, okay? We might meet for 2-3 times over this month to discuss the review paper. As we said last time, we might meet to present something about how to have a nice presentation. It's really not easy task. You need to take careful of the details. Last time I told you there is very important point. Most of you miss it in their presentation, which is slide number. Slide number. Most of your presentation won't have slide number, and that is a real problem. I told you last time, if you are presenting to someone and that one has a question, he will ask, can you go back 2 slides? Oh, no, the previous one. No, keep going. No, not this one, not this one. It will waste a lot of time by not having slide number. But if you have it, they will tell you, can you go to slide 20? How about this point? How about that point, right? And this is a good way to communicate and share ideas, collaborate, rather than wasting time on something not relevant to science, right? There are a lot of tricks, a lot of things that you should really pay attention to when you do the presentation. I might present about all those points within this month before your presentation. But before that, we really need to decide on the presentation day. Okay? And I know you'll focus on assignment number 3 and assignment number 4 next, right? So you might have about 2 to 3 weeks to wrap up your review paper and prepare the slides. Okay? So please do that as soon as possible. You will hear from me over the next week since we don't have lectures, so I have more time to go over your review paper, the first draft, and try to give you comments. If you made a progress more than what you submitted, please do submit it again, okay? So I can give you a comment on the updated version. I'm sure it's been a month or so since the first draft. Some of you lazy, didn't do anything in the last month, get busy with other courses. Some other might already did great progress. So if you do so, please resubmit it again, okay? And that's again for your benefits. So I would give you comments on the critical thing rather than the format of the slide. All right. Okay. Thank you so much for today. Do you have any question? Not really until you hear back from me. Okay. Hopefully before Friday or on Friday. If you didn't hear back, then there is no meeting. All right. But please consider that time to submit the assignment, work on the review paper. I don't want you to delay it until the end of the month, then you'll be in trouble. Okay? Any question?